Welcome back to ARM Studios. I'm Jason Foster, the CEO of Ori Biotech. Joining me now is Matthew Hewitt. Matt is the VP and CTO of the Cell and Gene Therapy and Biologics team at Charles River Laboratories. Welcome, Matt.
Thank you. Thank you for having me.
Thanks for coming. I think we wanted to talk to you a little bit about digital and digitization in cell and gene. You've had a long career in the industry. Maybe you can speak a little bit to your kind of career and how it's sort of maybe changed a little bit within cell and gene. And then we can talk a little bit about why we think digital is a priority for the industry to think about today.
Right. So when I started on the AAV gene therapy side, I like to say that I was working with a group at Penn and running the immunology part of that journey for AAV. And we were generally supporting about half the clinical trials in the U.S. at the time. And I always say that's less of a brag and more about how early we were in the field in this overall journey, and that the number of clinical trials in the U.S. were still quite small. Back then, we were all paper-based. We had a fireproof separate room and fireproof cabinets for all of our records. And then obviously once they reached a certain age, then they went off site for storage. That became a really big focal point for us at some point as we started to grow the number of programs that we were supporting in clinic was, how do we support this on a paper level? We were constantly having to scan things and send them off to clinical CROs, to the sponsors that we were working with.
Yep.
And we just knew that it wasn't scalable.
Yeah, it just becomes untenable after a while.
Right.
Yep.
And so, then moving into cell therapy, I don't know if it's any better. Certainly in the case of autologous, it's probably worse.
Yeah.
Because, we're producing, on average, I think as an industry, several thousand doses per year.
With thousands of pages of paper batch record associated with each. So.
And I think the thing that sometimes gets lost in this is when you do this at a CDMO, whatever the number of pages that you have in your batch record, actually you need to multiply it by 2.
Yes.
Because one needs to be reviewed by the CDMO QA, and then mostly in parallel, needs to be then reviewed by the sponsor QA.
Right.
So if you have a 500-page batch record as a strawman, it's really 1,000 pages.
Yep.
And then if you want to do 1,000 patients per year, then you're talking about 1 million pages of documents to review per year.
Reviewed by multiple people in multiple locations.
Right. It's a lot of trees.
Yes.
So we feel that there are different roadblocks or different challenges within cell and gene therapy, and sticking with cell therapy and autologous, our primary challenge right now is manufacturing.
Yes.
Right. So there's this famous quote from Christy Shaw about 2 out of 10 patients that are eligible actually receive a cell therapy.
Yep.
And there's some others that suggesting it's maybe 25% actually do.
Yep.
But the point is once we fix the manufacturing problem, we will. The next problem will be QC. And I'm sure we will come up with some very good solutions around that. And then ultimately the last problem, maybe not the last, but the next problem will be QA in that review.
Yep.
And if we're having to review things in paper and in person, we're just never going to be able to scale.
It'll always be a bottleneck, no matter how well we do that upfront piece.
Right.
And that, I think, is ultimately the topic du jour for us. Is really, we know that companies, when they're developing processes or products themselves or with a CDMO partner, they're often very focused on the science. We want to get the science right. We want to get the clinical efficacy, the safety right, and rightly so. What we see is that historical, singular focus on getting into the clinic, getting first-in-man data, hitting that inflection point, getting further along in your clinical process has been the formula for success in biologics or small molecules. We have followed that same formula in cell and gene and found that it doesn't really work as well in this new modality. So I'm interested in your thoughts on, how should we think differently about this new modality, about cell and gene? And, my argument I made in an article I wrote for Cell and Gene recently was we need to think about manufacturability, cost of goods, commercial viability at the R&D phase, at PD when we're developing our processes. Because when you get to phase 2, it's too late. You've already, closed too many doors and too many options to get to commercial scale. So how do you think about that from your position at Charles River and the work that you do, and just as a industry thought leader? What's your perspective?
I agree with everything you said. First of all, I think that I think we have to, as a field, and when I say as a field, I mean tools, CDMOs, and the therapeutic developers. We have to get really pragmatic about what we're going to take on.
Yeah.
And I mean, the therapeutic developers need to focus on therapeutics.
Yes.
That has to be their job. I think we have to move past this era where the therapeutic developers in this space, specifically in cell therapy, believe they have to have everything internal and everything has to be built by them.
Right.
I think that is something that is not scalable.
No.
And I honestly think, I understand, and we talk about this a lot in various settings about how do we standardize stuff. And I think we have to be careful and pragmatic about how we go about standardizing things, because as you start to standardize, you're going to limit innovation. So you want to do it in a way.
Flexibility.
Exactly.
But, yeah.
So we don't want to be with the same box, but I think there are areas that we can do it.
Yeah.
And I think there are areas that will help us quite a bit. So if the therapeutic developers focus on developing the best therapeutics they can, hopefully I think the therapeutic developers and the VCs and the other funders in the space have become much more savvy. They now understand that it's not only good to have good clinical data, it's not only good to have a BLA or MAA approval. You actually have to have a product that's commercially viable. And that's where the COGS come in,
Yep.
and that's where scaling comes in.
Yep.
And so I think it's incumbent upon, folks like Ori to work with folks like Charles River as a CDMO to figure out where we can help and where we can standardize. One thing that we talk about a lot is, electronic batch records.
Mm-hmm.
Certainly that's going to hopefully relieve some of the challenges and bottlenecks around, manufacturing, QA, and QC. I think one of the other things that we need to work on is kind of the typical logistics backbone and tracking, because that somewhat has to standardize as well. Because right now, if you look across the spectrum, a lot of the commercial products have their own collection methods for material. They have their own shipping requirements. They have their own, certainly, manufacturing requirements, and then they have their own administration requirements.
And most of it's redundant. It's all reinventing the wheel every time.
And so what the clinical centers end up having to deal with is half a dozen different systems.
Yep.
Multiple tablets and or computers,
different portals,
multiple different labeling
yeah,
systems.
Exactly.
And I mean, at the end of the day, that's going to limit adoption. So we've got to figure out a way to segment this and say, okay, this is going to become somewhat of the industry standard, and we're going to go from there. And then everybody goes into it. And we were talking about it in the case of hospitals. Really, if it doesn't plug into like Epic, right?
Yeah.
You're not going to get it.
Yeah,
They're just not going to do it.
You're locked out, yeah.
Right? So, I mean, we have to get pragmatic about this and segment it and operationalize it.
Yeah.
The saying I like to say is, we were in the Wild West back in the mid-2010s.
Yep.
We had these first approvals in 2017. Now we're to a point where we have to get out of the Wild West and get into the Henry Ford era. You have to operationalize things.
That's right.
It has to get on an assembly line, and we have to be able to crank these things out because there's a lot of patient need that's still out there.
Yeah. I think if Ori becomes the Model T for cell therapy, we've done our job well.
There you go.
But
it comes in any color as long as it's black.
That's right. Yeah. So they're not faster horses though, so that's good. You mentioned e-batch records and kind of digitization as an enabler of scale and throughput and standardization and consistency, all those things that we're striving for. One thing that we didn't necessarily appreciate the benefits of, Ori is an IoT cloud native platform, so you can kind of see what's happening in real time. You can intervene if necessary, but also just cycle times, the amount of throughput that you can get through a clean room, through a system. When you do a wet set of experiments and you download the data and you put it into Excel and you create some charts and you discuss it with your colleagues and you put it in some graphs and you put it into a PowerPoint, it takes you 2, 3, 4 weeks to figure out what happened, formulate a new hypothesis or a next set of experiments.
Yep.
We're cutting that cycle time down to 1 week or 3 days so we can get a new experiment up and running. It just improves the throughput of the entire system because it is standardized. We have the metrics we need. We understand what to do. And that sort of is digitally enabled. The only way to do that is, if you're on paper and you're reading someone's lab notebook scratchings or, trying to download unstructured data out of a spreadsheet or 6 different spreadsheets, even worse, trying to do a root cause analysis. If there was deviations that you didn't understand the root cause of, it's impossible. It takes months and months. So, I think it's underappreciated that how hard we've made it for ourselves in the current environment.
Yeah.
Being very analog, very paper-based, and
I
not adopting these things.
The point I make a lot about that is a lot of people say they're very disappointed in the lack of progress we've been made. And I will say I can't take issue with that because prior to 2017 with no approvals, how many people were actually willing to go in and risk the development of new tools in this space? I mean, it just wasn't going to happen. There was too much risk. Now we know that this is a durable field, and now we're seeing a wealth of technologies come out.
We are.
I think the technologies have to work together, obviously, and we're taking a pretty pragmatic approach to this at Charles River, where electronic QMS systems, electronic dock control, MES, LES for QC and LIMS, all coupled together provide the systems needed to scale at commercial, withstand the regulatory scrutiny. But I think as well, we've seen that there is indications from FDA, from others, that decentralized manufacturing is coming. And so the only way to really do that is with electronic systems.
Yep.
This is always a can we or should we? Can we do this with paper? Yes. Should we? No.
Yeah.
I also think the IoT stuff I think one of the main things that we've struggled with, specifically on autologous, has always been the patient variability.
Yes.
And the question I think some of us have been asking in manufacturing for some time is, are we locking our processes incorrectly? Because the traditional method of locking your process is based on schedule. Day 1, I do this. Day 2, I do that, and so on and so forth. That works when you're dealing with a standard cell line that is very well predictable.
Standardized inputs into a standard process.
When you look at patient-to-patient variability in autologous, the question becomes, should we be locking processes based on metabolic activity, based on cellular activity?
Right.
And is that why we're seeing some of the failures that we're seeing in manufacturing?
Mm-hmm.
The only way we can do that is with the process analytical technology integration coupled with IoT.
Yes.
Right? Because we need to be able to see it in real time. Otherwise, how are you going to make adjustments on the fly?
Exactly.
And I think that opens up a whole new world for us in terms of manufacturing.
I mean, the way I talk about this, and I know far less about this than you do, is the GMP, to describe it in simple terms, we validate that we put the same input, we do the same process every time, and then we validate we get the same output. That's traditional GMP manufacturing. Right. In cell therapy manufacturing, we put a variable input into a standard process. And then we try and say we get the standard same thing out every time, and no, we don't, because that's not the way it works.
Yeah.
So, this idea of doing adaptive manufacturing, which is what, I think the logical endpoint of what you just described is actually what we do today. When you do deviation management,
right,
we're using human beings that are highly expert to say, this happened, now what do we do? We call that a deviation today, but actually we're adapting to what we see in the process to try and manage to that same endpoint, because that's ultimately what the FDA wants. That's what the therapy developer wants, is to get the same safe and effective medication out the other end. And so we do what we need to do in the process to get there. And that's effectively adaptive manufacturing. But if you have the data to support automated intervention or remote intervention or standardization across a network, that's really the only way to get there.
Yeah, we're already inching our way in that direction, right? Because in your system, as well as in other systems like the Cocoon that I worked on in the past, we're doing things like we're just deciding when we're going to do media exchanges or additions.
Yeah.
And then nobody actually approves that. It's just done, right? And the way that we've typically dealt with CQAs in the past for the variable manufacturing is you set them in a way that they're readily achievable from what you have in front of you. Now, we're probably going to see eventually FDA and others try to start holding our feet to the fire a little bit on this.
Narrowing those parameters a bit.
Yeah. Right now you can get away with a minimum transduction efficiency of 10% typically on a car product. They may decide that eventually that's just not going to do it and we want to raise it.
Yeah.
And the only way we're going to consistently be able to release product is if we do away, and we with a lot of the manual stuff. And I think as well as you're going to train any 2, 3, 4, 5, 50 operators, if you replace many of those manipulations with a system, even if it's something as simple as media additions or exchanges, you're going to get far less variability, which just means you're going to have generally a more stable and robust process.
Well, you solve two problems at once, right? So you standardize your process, you standardize the amount of variability, or you try and reduce the amount of variability, but also you don't have to hire so many people, or those people can do other things that are value added versus more rote kind of tasks. Because throughout this conference, this week people are always talking about the talent shortage and where we're going to find these people. If you want a bunch of PhD immunologists to work in a clean room 8 hours a day, 5 days a week, it just doesn't happen. That's not why they went and did it.
The other thing to consider is that by some estimates, population will peak in the next decade here in the U.S. and then potentially begin to decline or at least stay level. Mm-hmm. So as we continue to try to scale, we're not going to have a even larger pool of people to pull from in the future. So if we're going to do this, we either have to open it up make it more accessible to lower credentialed
Yeah.
folks, and or make better use of the folks that we have.
Yeah, and just reduce the total demand for labor.
Right.
I mean, that's the way, if we automate certain processes, we can reduce the amount of labor we need by, we think in our Gen 1, maybe by 50%. So
Right.
you need half as many people, and the people that remain could be technician level, maybe, high school graduate educated versus, master's or PhD or college educated. And that just opens up your talent pool
Exactly.
to be able to recruit from.
And the other thing it does, if it's closed, right, which you are,
Yeah.
is that you can go to lower grade classification clean room space. Now your gowning requirements change, which do two things. It saves you money, and it makes your people a lot happier. Because if you've ever gotten, bunny suits are not, the bunny suits are not enjoyable.
It is not. It's very hot and very unpleasant. So you mentioned flexibility and kind of interoperability. So this idea of technologies having to talk together and work together, a lot of, historically, technologies just haven't been thought of in that way. They're sort of a standalone piece of technology. It doesn't necessarily interface or link into other systems. We've tried to take, let's call it an open architecture approach and an ecosystem approach. So we link into systems like a Benchling or a TrackCell so that the data can flow to where it needs to go, maybe into an e-batch record system or an MES system or other types of systems, wherever your team might need them to be. And that, I think, open architecture, API-based connections and connectivity is well explored in technology everywhere outside of our field. I think that, for me is kind of the minimum threshold requirement, right? I mean, it has to be.
I'd hope so. I think there's a balance in that, and what we've dealt with in the past is, in trying to build connectors, is that, dealing with GXP software that's Part 11, Annex 11 compliant, if we start to get too crazy with the APIs, we don't want to invalidate that.
It has to be secure, has to be safe,
Right.
It has to be GMP compliant.
And I think we're going to be dealing with some different challenges in the future on this as well. So there's kind of two solutions. One in the near term is we can certainly use middleware, and there's several companies that do that, that stick it out into a certain place in a location within the limbs, and then we can have crawlers go and grab it and compile them. I think on the longer term, one thing we're going to have to deal with is that, right now, a lot of the systems we use have, a piece of hardware in itself and typically a separate computer, right? So it's for a lot of corporate security, that's much more reasonable to them because we can install our software security and everything else. And I think the future is not going to be that there's two separate boxes. I mean, it's going to go fully embedded.
Exactly.
And so, we have been working with Charles River IT as an example. We have a very good IT support group, and to say, look, this is not sustainable to continue to think we're going to be able to put our software onto each embedded system, because you will invalidate much of the compliance. So how do we do this? How do we create pipes for these? How do we do manufacturer assessments, on whether or not they're suitable to go into our systems? And then, keep them in somewhat of a sandbox so that they don't potentially do something silly if they are infected with some spyware. But, we're going to have to take a risk-based approach, which is what we do in general.
Yeah, for most other areas. And one of the way we're also trying to solve for this is we could embed the control system essentially for the Ori platform into an automation system, a SCADA platform, Delta V, Ignition, some of these that would embed in your system. So you don't actually need a laptop sitting next door or whatever.
Right.
It already would be in your validated, automation control system.
Which makes so much sense, especially when we're talking about scaling.
It's really
if you look at what
sense.
all of us envision as the ultimate scale, it looks like NORAD, right? So you're sitting in this big room,
wall of monitors.
Monitors. It's telling you exactly what's going on across, 200 Oris or 200 Prodigies or whatever. The point is that's the way it's gonna really make sense. Because the other way is basically you have folks roaming your suites and then manually checking if there's problems.
Right. Staring at small screens in clean rooms, which doesn't make any sense. So.
Exactly.
Well, Matt, thanks so much for joining us. And I was going to say welcome to the Ori Scientific Advisory Board. Thank you for joining. I'm excited to have you as part of the broader Ori team and look forward to working together in the future.
Yeah, same. Thank you, Jason.
Thanks so much.
Bye.